Welcome everyone to the ChBE Student Team Showcase, which is part of the larger ChBE Student Conference this year. My name is Brandon Chen, and I will be sort of the moderator of this event, if you will. So the aim of the Student Team Showcase is really just to give a brief introduction to some of the student-led teams here operating in the ChBE department, and just to kind of introduce each other to what we've been working on over the past couple months. So in terms of format, each team will be presenting a roughly 5-minute or so presentation, 5 to 7 minutes about their team. The presentation order for today is first UBC Envision, then UBC BioT, UBC ChemEcar, followed by UBC GreenJoule, and last but not least, Best BIT. And to start off, I think it would be great if we could do a quick round of introductions. So I guess I can go first. I will be presenting on behalf of UBC Envision, and I'm currently serving as Envision's president. Do we have anyone from BioT here?
Yeah, hi, I'm Keegan, team lead of BioT. Ken is also here. He can introduce himself.
My name is Ken. I'm from BioT as well. I'm the Brewing team lead of BioT.
Perfect. Thank you. Thank you. Anyone from ChemEcar?
Hi, I'm Nika. I'm the co-captain at ChemEcar. And with me, Noor, would you like to introduce yourself?
Yes. Hi, my name is Noor, and I'm a first-year student in the lab team of ChemEcar.
Perfect. Thank you. GreenJoule?
Hi, everybody. I'm Monse. I'm one of the co-leads of GreenJoule along with Daniel.
Hey guys, I'm Daniel. I'm one of the other co-leads for GreenJoule. Nice to meet you guys.
Perfect. Thanks, Daniel. And then certainly last but not least, Best BIT.
Hi, I'm Sam here, and I'm one of the members at Best BIT, and I'm here with Hannah.
Hi, so I'm Hannah. I'm also one of the members.
Perfect. Thank you. All right, let's get right into it. So as I mentioned before, my name is Brandon Chen, and I'm serving as the current president of UBC Envision this year. Just a quick note, I am currently in my 5th year at UBC in chemical engineering. And I think we also have Mateus on the call, I believe, who is our vice president. So if you are here, Mateus, would you like to give a quick intro?
Hi, everyone. I'm Mateus. I'm the VP.
Perfect. Thanks, Mateus. If we have any other Envision officers, I don't think we do. Quickly looking at the participant list. We've got a couple other officers on board as well. Okay, so first let me introduce you all to UBC Envision. So Envision really has kind of 2 main parts and 2 main functions. So the first is to act as the student chapter of AICHE, or the American Institute of Chemical Engineers, here on UBC Vancouver campus. And AICHE is the leading professional organization, comprising of students, professors, and industry people alike, all in chemical engineering related fields. Although AICHE is technically an American institution, really there are members from all countries all over the world. So it's a really good chance to network with people really from outside of North America in the larger chemical engineering world. The second part of UBC Envision is to act as a project incubator within the Chemical and Biological Engineering Department. And currently we support 3 other engineering student teams, which you will hear from later on in this presentation. We also welcome any student looking to start up a project within their department, or specifically our chemical engineering department, to get in contact, and we can help with the administrative support and getting your project up off the ground. Officially, we are an ex officio club under the Chemical and Biological Engineering Department, but we do welcome all students from any discipline to join our teams. So here's a brief list of some of the events we have hosted and a couple of the events we are currently planning for the future. So this year, we've launched a new speaker series event where we've invited professors, industry people, and a couple grad students just to talk about their experiences, their research, and what they've been doing over the past couple of years, and also to provide useful tips for students' professional development. So we've hosted 4 of these speaker series this year so far, and we have one upcoming one, I believe next week, with an organization called Casa Congo. So keep your eyes peeled for that. Relating to our role as an AICHE student chapter, we also have supported students attending the national AICHE conference back in, I believe it was November. And we are currently collaborating with ChemEcar and planning the 2021 Pacific Northwest Regional Conference for 6 universities in our local region. And so both conferences featured various networking and professional development events, as well as research presentations and the famous Chemi Jeopardy and ChemEcar competitions as well. Additionally, we have hosted several meetings with our AICHE sister chapter at UPC, in Barcelona, Spain over the last year, and we look forward to more collaborations with them in the future. Finally, as I'm sure you're all aware, over the last academic year, we've been helping organize this CHBE Students Conference. So hopefully you're all enjoying Industry Night last night and Research Day today. So that concludes my short introduction to UBC Envision. If you would like to find out more about Envision or simply to hear more about our events, please follow our social medias below on the last part of our slide there, or check out our website. The 3 projects that we support can also be seen above, but they will be all presenting shortly.
Hello, everyone. It's Ken. And I'll firstly give an overview of the team BioT, which stands for Brewing Internet of Things. The 2 focuses of the team is to implement a pilot scale brewery to enhance students' engineering education to applied learning, and developing advanced technologies focusing on the concept of Internet of Things, which can be applied to real local breweries. The team develop craft brewing technologies and we make efforts to create an automated brewing system with the ability to monitor and control process variables such as temperature, acidity, and specific gravity in real time. We make we try to design and implement low cost data monitoring and have our innovative techniques and advanced technology be applied to actual breweries to brew perfect beer through various projects. And it has great potential for microbreweries or home breweries because they cannot afford the high-end expensive equipment like the bigger breweries. Yeah. And when in-person work was still allowed, the team would conduct collaborative brewing sessions with actual local breweries. Once everything's online, now we still try to connect with breweries to learn their modern brewing technologies and technical skills by having online conversations with them. Next page. And I'll give a short introduction for the brewing subteam. The brewing subteam, its main goal is to apply the theories taught in CHBE courses to develop a bench-scale brewery. The team develops methods to optimize the brewing process by analyzing the product and the intermediates during the fermentation process, and creates new recipes through research and determine the conditions needed for the consistent product quality. The brewing team collaborates with the lab and instrumentation team to research the reaction kinetics in the fermenter and test the key characteristics of beer, such as bitterness, alcohol by volume, and the standard reference model, which is color of the beer. Some current ongoing projects are listed here. The first one is sustainable brewing algae beer, and the goal is to conduct carbon capturing with algae during the fermentation process. The goal is to bring awareness to climate change and promote sustainable technologies. The team got in contact with an Australian brewery called Young Henry's Brewery and a biotech lab at University of Technology, Sydney, who are currently working on the same project on a bigger scale. So we are looking for future collaboration. There are also yeast recovery repitching project to optimize the brewing efficiency and cost, and the water analysis additive chemistry project to study the effects of different salts, the color, flavoring, and other properties of beer for further developing new recipes. And now I'll pass it on to Keegan to talk about an instrumentation team.
Thank you, Ken. So I oversee the instrumentation team at BioT. The instrumentation team, their goal is to build and design analytical equipment to monitor and control the brewing process variables. As Ken mentioned before, our goal is the steady state automation and data capture. So the instrumentation team does their heavy focus on is getting that part off the ground. But so their ongoing projects are the process automation, and this year we're looking into developing a programmable logic controller, or a PLC, with a user interface capable of displaying data from pumps, fermenters, and tanks. And then that's going to be during the brew day session. So it's going to cut back on a lot of the manual labor that a lot of the brew team has to do. A lot of stuff that is inaccurate to do by hand and can much easier be controlled by a computer is a lot of the heading of that project. And then secondly, they're also focusing on the data collection during the fermentation process, which as you can see is not on the figure here, but this is a brew day figure where after this, the brew wort is the technical term, would go off into a fermenter, and we want to collect that data because that's going to allow us to make changes into our recipes. It's going to allow us to understand when the brew is done and maybe see if something went wrong. And then they're currently working on integrating current brew lab code with BioT's new needs. And that's done on a Arduino, a set of Arduinos, and possibly in the past it has been done on a Raspberry Pi as well. Next page, please. So our last team in BioT is the laboratory team, and their main goals are to create their main goals are to analyze the data collected by the instrumentation team, and they try their best to create data clusters and develop models to process the data and understand the data. And then once that understanding gets finished, that's passed on to the brew team and to make standard, to apply the knowledge that they have now analyzed. Some of their current projects are last year. This is actually a project from last year, but we were looking into the genetic modification of yeast using the CRISPR-Cas9 system to create glow-in-the-dark beer. It was a very interesting project done during the week of eWeek, and was looking to be applied within eWeek, although we didn't have the current tools and resources in order to do it. But it's something that would be very interesting to do in the future, and it's definitely still on the front foot of BioT. Another interesting thing that the laboratory team can go into is protein beer. And then this is more on the educational side of things, where you're kind of getting out of the traditional brewing practices and you're going into more specific brewing controls, something very unique, not often seen. And then another very important thing that the laboratory team does is they measure variables that the instrumentation team cannot. Whereas there's not sensors for IBU and SRM, which stand for International Bitterness Unit and Standard Reference Model, deterring the bitterness and the color of the beer. But something that the laboratory team can do is use their chemical engineering knowledge and develop the models for measuring that. And that's put in place for developing SOPs as well, something that the laboratory team takes head of in BioT. Yeah, something else that's interesting about BioT is we hold weekly or bi-weekly beer reviews. And that really helps the team get more knowledge of beer in the first place. It's great hands-on experience, and we're having a great time doing it. It's very, very technical. It's not totally set back, but it's one of the more fun parts of the team. And then on to the last slide, please. So that'll wrap up BioT for today. If you have any questions, I don't know how Brandon wants to arrange that, ask them now or ask them at the end. But if he wants to let us know if we can take questions now.
Yeah, if anyone has any questions now, we've got about 2 minutes or so for people to ask questions. Again, we can have more questions at the end, whatever people are kind of comfortable with. So if anyone has questions for BioT, feel free to, you can unmute yourself and ask, or ask them in the chat, whatever is easier for you.
Did you guys manage to make the glow-in-the-dark brew?
No, we never did the genetic modification. The picture that you saw was actually using vitamin B12 vitamins under fluorescent light. It glows when we were looking into different concentrations of that, and if that would actually work within, if that could actually be applied. But the genetic modification never got off the ground due to we needed the proper laboratory, and we ran out of time as well. It was a bigger factor.
I was just curious what that would have tasted like.
Yeah, we actually got told a couple times that it might not be safe for human ingestion. So that was also another main factor. But I would love to still be optimistic in that you could still drink it. But who knows? That's for future problems.
That's a living on the edge kind of drinking beer.
Yeah, it really is.
I would do it.
I would like to do it if it was safe.
Alrighty, any other last questions for BioT?
I actually had a quick question for the protein beer. What protein did you really add? Like, was it some kind of mutation you did that added the protein, or was it some sort of mix-in that you added in?
I believe I haven't overseen the project in a couple weeks, but I believe the modifications are done within the malting process and kind of the ingredients that you add into the beer, changing up the wheat and the grist ratios within that first barley count. I believe is where you get a lot of the the forward protein. It's hard to explain, but yeah, it's I believe it's not through a mutation, it's through an ingredient adjustment.
Hi, everyone. It's Nika. And again, I'm one of the co-captains at ChemEcar. I would definitely love to see that glow-in-the-dark beer happen. But now onto ChemEcar. Next slide. We are an engineering design team and a competition-based design team, which is hosted by the American Institute for Chemical Engineers. And our goal is to design shoebox-sized cars which operate on chemical reactions. And the car has to travel an undisclosed distance on competition day with some weight attached to it that we're not given prior to the competition day. And it's a multidisciplinary team. We have 4 sub-teams, which I will talk about shortly. And it's a great way to kind of learn about all the aspects of chemical engineering and some other disciplines as well. Next. Another one. So our first sub-team is the circuitry or electrical sub-team, and their goal is essentially to build the electrical systems that are involved in starting and stopping the car. They manage the sensors and data collection for the stopping mechanism, and as well, hooking up the battery to the car, and, the motor and everything like that. So they're kind of the team that puts everything together at the end and makes sure that the car can run. Next slide. So right now, the circuitry team is working on some Tinkercad. So they have been modeling their circuits online last semester, and then this semester, they are working on some robots. So we ordered some kits with all of the stuff that they require, and they're making some robots, and they're looking into developing an app to control the robots to drive them. So they're getting a little bit of circuitry experience through there that they can hopefully apply to ChemEcar once we're back in person. Next. So we also have the mechanical sub-team, and they have to build the actual vehicle. So they work with battery and the stopping mechanism team to develop casings for the batteries and for the stopping mechanisms. They deal with building the chassis and the wheels and everything to put together. Their main challenge is to fit within the parameters, the physical parameters of the sizing of the car, and also to make it light enough to be able to carry everything, but still actually move. Next. Thanks, Brendan. Right now, the mechanical team is doing some awesome work. This is one of the examples of their work this year online. And every member of the mechanical team has built like battery casings for potential batteries that we will use in the future. So you can see the assemblies here. They've also worked on all the other components of the car. This is just an example. And it's really great to see everyone be able to, get ready for when we can actually build a physical car and improve their skills even in an online setting. So next we have our battery team or our power source team. One of the aspects of ChemEcar is that we have to build our own batteries. So we can't use, Duracell batteries to hook up our car. And they essentially have to build them from scratch. Think about, will it be efficient? Is there too much internal resistance? Things like that. And they also have to work with the mechanical team to develop the casing for the battery in order for it to work properly. Next. And these are a couple examples of batteries that we have researched this year. So the main goal for the battery team this semester is to research and create a database of technical reports that they can go back to in the future and develop those batteries which they were interested in researching. So we have a lithium iron phosphate battery, a thermoelectric generator, magnesium air. They've been working on some other ones as well. This is just a couple. And it's really great to be able to, research in depth the batteries that we will be able to build in the future. And it's a great experience to learn a little bit about electrochemistry outside of the classroom and really apply it to something real. I believe Noor will present the lab team. She's one of our new members on lab.
Yes, so the laboratory team is focusing right now on researching reactions that have an identifiable change, either temperature, color, pressure, which the circuitry team can identify and therefore create a system where it could code for the braking system to happen. So these past few months, we've been researching different reactions. And on the competition day, we're given a certain distance that the car needs to travel. And with the mechanical team and the circuitry team, we work together to determine the distance and the time required for the reaction to occur, as the time of the reaction will indicate when the braking system will actually happen. So in the next slide, these are just a few of the reactions that we've been working on. So we've currently made, each pairs, we've created a few technical memos that we're going to hopefully come back to when things are in person again. And we've done the sulfur clock reaction, ion exchange resin bead reaction, and sodium hydroxide and copper sulfate reaction. And one that's not listed here is the iodine clock reaction, which a bunch of us found very interesting. It's when triiodide reacts with starch to create a very dark colored compound. And it's interesting to study the different reactions and see which ones have a bigger change in quality that can be identified better. So hopefully when everything goes back in person, we'll be able to experiment and see which reaction is actually going to hopefully work for us on competition day.
Yeah, so that's ChemEcar. We have our 4 sub-teams that are currently researching, developing their skills, and then we are hoping to participate in the regional competition that's happening at the end of April with an actual physical car. So we're getting our safety documentation ready to be able to do that. And we're just really excited to be able to hopefully build a ChemEcar soon with all of the research we've done this year. If you're interested, our recruitment happens in every September, and these are all of our social medias and email and website if you would like to learn more. Thank you.
Perfect. Thank you, ChemEcar.
Okay, hi everybody. I'm Monse. I'm one of the co-leads of GreenJoule this year, along with Daniel. And we're really excited to be here today and have the opportunity to share what we've been working on in our research for the last couple of months. Next slide, please. Okay, so what is GreenJoule? GreenJoule is basically a student-led research team which works under UBC Envision, and our goal is to optimize the production of microalgae-based biofuels by simultaneously producing biodiesel, and we're hoping to expand that to bioethanol as well. Next slide, please. Okay, so our research. Obviously, GreenJoule throughout the years has conducted extensive research on microalgae, specifically Chlorella vulgaris. And these are just some of the advantages that we have found and why we believe it can be so useful. So first of all, and most importantly, microalgae biodiesel has the potential to alleviate global reliance on fossil fuels. In addition, it can really help to reduce the greenhouse gas emissions by removing carbon dioxide from the air, since it is considered a photosynthetic organism. Additionally, algae does not have any specific growing conditions, and it doesn't compete with agriculture for land or for fresh water. Furthermore, microalgae can actually clean wastewater. And wastewater is a cheap nutrient source, it's abundant, and it turns into clean water after the growth cycle has been complete. In that way, the process economics can really be improved significantly by contributing to wastewater treatment. And lastly, microalgae has a high lipid and carbohydrate content, which is really key in the production of biofuels and bioethanol. But with that being said, there is obviously a big debate about whether or not biofuels are actually carbon neutral. And you'll hear a bit more about that through our impact analysis subteam. But these are just some of the potentials that we have seen with microalgae, but there are obviously some big strides that we have to make in order to fully optimize the process before we can achieve these results. And now I'm going to pass it along to my co-lead Daniel to talk a little bit more about each subteam.
Hey guys, I'm Daniel. I'm one of the other co-leads for GreenJoule. So here's a brief overview of our 4 sub-teams. We have the growth team, which focuses on researching and cultivating the algae strains and figuring out the suitable conditions for the specific algae strain that we're growing. And then next we have the extraction team, which focuses on finding the best extraction processes for turning the biomass into biofuels. Then thirdly, we have the monitoring subteam, which focuses on monitoring and collecting the data on the growth parameters and making sure that these optimal conditions are consistent and stable. And lastly, our newest subteam is the impact analysis team. And they focus on qualifying and quantifying the environmental impacts that biofuels in general, and then more specifically algal biofuels have on the environment. So yeah, let's head on to the next slide. Right, so we're going to talk a bit about the research that we've done over the past few months. So I'll be speaking on behalf of the growth team today. So we're working towards growing an algae strain called Chlorella vulgaris, and we want to find the optimum conditions to increase the lipid yield for production of biodiesel and bioethanol. And we also want to be able to treat the wastewater in which the algae grows in. So the algae is to be grown in a photobioreactor using a growth medium called Bould's basal medium. It's essentially a combination of ionic compounds that aids the algae in its growth. And then in addition to that, the light intensity, the carbon dioxide concentration, the temperature, and the pH, which are just a few of the parameters that the algae grows in, these things are varied and they're monitored until we find the optimum conditions that favor the growth of the algae. And once this process is done, the algae is harvested through microfiltration using a polyvinylidene membrane, and then we'll be able to obtain the biomass of the algae. So yeah, let's hand it off to Xavier from the extraction team.
Hi, so I'm Xavier. I'm a member of the extraction team. As Daniel said, the extraction team's goal is to find the best method of obtaining the biofuel from the algae that we receive from the growth team. Several methods were compared during this year, and we found that the best method of extracting the biofuels was from hydrothermal liquefaction, which is found to be the most efficient of these methods. In this process, biomass is treated between 520 and 647 Kelvin at 200 atmosphere. These moderate temperatures and high pressures are used to break down the biomass into its smaller and liquid components. And the water and an alkaline catalyst are added to the biomass to utilize a pyrolytic mechanism. And a pyrolytic mechanism is just the use of high temperatures to decompose an object of interest. And in the case of this project, it will break down the biomass and allow the biofuels to be separated easier. Next, Puneet will explain the monitoring developments this year.
Hi everyone, I'm Puneet and I'm a member of the monitoring team at GreenJoule. The monitoring team at GreenJoule is trying to optimize algae growth through the tracking of important growth parameters. Our goal is to help control and monitor the various aspects of the algae growth medium to help increase biofuel yield. This year, we collaborated with the growth team to determine the most important parameters of the algae growth medium, which were the pH and the temperature. Currently, we are working on programming an Arduino and various sensors to help monitor these variables. In addition, we are developing a process control mechanism for the pH of the growth medium, which aims to maintain the pH between 8 and 9 for optimal growth. Finally, we hope to integrate our Arduino and the sensor setup with a Raspberry Pi to create a dashboard to display real-time values of pH and temperature. The monitoring of these various parameters is important as it can give insights on algae growth and biofuel yield. Now let's hand it off to Marina for the impact analysis team.
Hi everyone, my name is Marina and I'm the team leader for the impact analysis subteam. So as previously mentioned, impact analysis is a new addition to GreenJoule. And obviously we know that biofuel will be less impactful on the environment than, for instance, using fossil fuels. However, how environmentally friendly really is the biofuel production process? And this is the question that the impact analysis team is aiming to answer. So in order to do this, we've been both working to qualify and quantify the environmental impacts of the biofuel production process. So we've been experimenting with different life cycle analysis software, specifically so far with GHGenius, and looking at all the aspects of the product's life cycle. So from its extraction to its usage to the end of its life cycle and what gets done with anything that gets left behind. We've also been working to compile relevant research as well as integrate numerous sustainability concepts into the project. We've been putting all of this into a report that we're hoping to use for years to come. Thanks so much. And with that, I'll pass it back to Daniel.
Thanks so much, guys. Yeah, thanks for listening to our presentation. And if you have any questions, feel free to ask. And we also have a QR code there. We just did a recent interview with the Weather Network, and they came in the lab with us and they asked us questions, and it was a really nice interview. So feel free to check that out. And also we have our contact info and social media on the side there. So thanks so much.
Okay, so hello everyone. So my name is Hannah and I'm here with my partner Sam, and we're both members of BIT. So we are part of the UBC Biochemical Innovation Team, which is a sub-team of the UBC Biomedical Engineering Student Team, which is UBCBest. If you could go to the next slide, please. So UBCBest is a multidisciplinary design team dedicated to creating innovative solutions to problems within the medical space, both locally and internationally. And currently BEST consists of 8 sub-teams, and as I said, BIT is one of them. And you can head to the next one. So
our team
go ahead, sorry.
Our team is a multidisciplinary sub-team, and we've changed since 2 CHBE members created BIT within BEST in 2018. They noticed that CHBE students lack exposure to design teams working in wet labs, and so the biochemical design projects were sort of launched. We were only a biochemical focused team, and we are the only biochemically focused team in BEST. Traditionally, more MEC, ELEC, and CPEN students were part of the team, but we currently have a lot of CHBE, BMEG, and ENCHFIS students. And as you can see, this slide shows our current members right now, and the other image shows us in the lab. You can move to the next slide, please. So BIT is currently undertaking 2 projects. BIT's first project was focused on developing a much needed alternative treatment for insulin resistance and potentially diabetes mellitus, also commonly known as type 2 diabetes. The project goal was to determine the effects of extracted cinnamon polyphenols on glucose uptake. So this project was divided into 2 parts. The first part was focused on polyphenol extraction using cinnamon. So our team was working on extracting and quantifying polyphenols from specific species of cinnamon. The second part was assessing the effects of glucose uptake in pre-diabetic adipocytes. So we wanted to test the extracted polyphenol and see its biological effects on mammalian in vitro models. So our team decided to use insulin-resistant fat cells, which are also called adipocytes, and assayed them with the concentrated polyphenol extract to determine how dose-dependent effects affect the cell growth and glucose uptake. As you can see in our slides, the pictures on the bottom show the flow of how the experiments were conducted. So I can walk you through it actually. So the first picture shows how our team obtained the raw cinnamon powder. The second picture shows how we perform sockets extraction using ethanol in the CHBE labs to extract the polyphenol. The third picture shows Travis, who is the lab manager at Kiefer Labs, and who is also our team guide, who's preparing a sample extract of a given concentration using DMSO, which was then fed to our sample cell line. The fourth photo shows how the first differentiation of the adipocyte fat cells look at day 8. This was taken at the Kiefer Labs using an EM microscope. As you know, due to COVID, we had to put this project on hold, and because we are not able to go back into the lab and perform more experiments. However, with the new guidelines coming into place, we will hopefully be back in person in the lab, hopefully during summer. And we will be taking the given measures by AppSci department, of course, but we're excited to get back with this project. You can move to the next slide, please.
Yeah, so as Sam said, we needed to choose kind of a different project because we can't do a lab project remotely. So as our remote project, we decided to tackle a bioinformatics project regarding coronary heart disease. So just a little background information on that. CHD occurs when the blood vessels that supply the heart become diseased and the heart does not receive enough oxygenated blood. So the problem we found is that not very many genome-wide association studies are very diverse, and they focus a lot on European populations. So we want to provide data about CHD for all different populations, including minorities. So our project hypothesis states that certain populations present specific CHD SNPs at a higher allele frequency than others due to genetic underpinnings. And we want to investigate the commonalities in biological function or metabolic pathways between commonly reported CHD SNPs. So we did spend all of last summer researching like a project topic, and we decided this one, and we are currently learning a lot about bioinformatics and machine learning. And our new member, who is actually a part of engineering physics, is kind of helping us with that aspect. And you can move on to the next one. So yeah, so that's basically a summarization of the 2 projects that we have tackled so far in our current project due to COVID. So for summertime recruitment, please visit the UBCBest Facebook and LinkedIn, because that's where we'll be posting job postings. And we are currently looking for CHBE members. However, if you know anyone that have the skills that fit the position description, please apply. And we also have our email and other socials listed at the bottom if you would like more information. And yeah, that's basically it.
I'd like to thank all the teams for being here first off. We appreciate you being here and showing off all the hard work you've been working on for the past year or more. So thank you to all the teams. Also, a brief thank you to the CHBE Graduate Student Council for helping organize Research Day today. It's been a big help. Also to the CHBE department for hosting this conference. And we hope those in the audience learned a bit about our student teams, and perhaps any students in the audience might be on one of our teams next year. And with that, I'll end the CHBE Student Team Showcase